Good evening, everyone, and welcome to our Scas Spotlight Speaker Series, highlighting the fascinating research conducted by our diverse board members. My name is Mia Adriani, and I am the current Scas president. We started this speaker series during the pandemic as a way to engage with our membership. And the feedback was so good that we decided to keep it going over the last few years. And I certainly enjoy it as well and learn a lot more about the board members and what research they do. So please stay tuned for our next Spotlight talk. We'll be sending them out and you'll hopefully learn about them however you learned about this one, and we'll do a couple more leading up to our annual meeting, which I'll tell you about in just a minute. Please check on our website for updates, which is scas.nhm, Natural History Museum.org, for updates, as well as a chance to go back and watch previously recorded Spotlight talks if you're interested. I'd also like to let you know about our upcoming annual meeting, which will be held at UC Irvine on April 25th of this next year. Registration will open in January. So again, please check the website for updates and deadlines and other opportunities hosted and funded by Scas. To learn a bit more about those opportunities, I'll turn it over to one of our board members, Kimo Morris, who will tell you a bit more about those opportunities, as well as introduce tonight's speaker. Feel free to use the chat for questions as they arise, and also comments if you'd like. And I want to send out a heartfelt thank you for supporting Scas over the years and your participation. Take it away, Kimo.
All right. Good evening, everyone. My name is Kimo Morris, as Mia mentioned. I'm also a member of the Scas board. My role on the board is to oversee our research training program, which pairs bright young high school students who are considering science as a potential career pathway with researchers, mostly in Southern California, although some mentors are actually outside of Southern California and in some cases as far away as the East Coast. In addition to that program, I would like to put a plug in for our 2025 SCAS student grant. If you are a graduate student or an undergraduate who is engaged in research, please consider applying for this competitive grant. Faculty members in the audience, please let your students know about this opportunity. For a list of past award winners and how those students utilize their award, please visit the SCAS website. Students, the only catch is that in the year that follows the announcement of your award, you are expected to give a presentation at the SCAS annual meeting and also write a brief summary of your work to our SCAS newsletter, The Smilodon. The current application is available on the website and must be submitted no later than February 15th, 2025. At our next annual meeting, which is being held at UC Irvine on Friday, April 25th, we will announce the winning grant recipients. So if you are a winner, you will be giving an oral presentation or a poster presentation at the following SCAS annual meeting in 2026 and contributing a short write-up for the newsletter. Okay, now on to the business at hand tonight. It is my pleasure to welcome my fellow SCAS board member, Dr. Craig Gelpi. Craig originally hails from St. Bernard Parish, Louisiana, and graduated with his bachelor's in both physics and mathematics from Tulane University. He then earned his PhD from the University of Houston, studying aurora and radiation belt processes. He did a postdoc at Boston University, where he analyzed magnetosphere processes for the Air Force Geophysical Laboratory. Craig moved to the LA area in the mid-80s to work on ballistic missile defense and other related topics. He's an avid scuba diver and was an active member of the Southern California dive club, the Sand Eaters. Throughout his professional career, Craig combined his ocean knowledge with his background in space and remote sensing to develop satellite techniques for offshore petroleum exploration. These techniques required an understanding of radiative transfer in the atmosphere and were used to interpret satellite observations of the ocean. He combined his expertise in space physics and ocean phenomenology to develop and test underwater navigation techniques required for long-duration underwater autonomous vehicle operation. He is now a chief scientist of the Catalina Marine Society. The society emphasizes research diving to understand ocean processes in the Southern California Bight. Craig has been retired since 2022 and now splits his time between Nacogdoches, Texas, and Metairie, Louisiana, but continues his work for the society, spending a good deal of time here in LA. Craig's here to tell us all about his 20 years of Catalina Marine Society ocean research at Catalina. To reiterate what Mia mentioned, throughout Craig's talk, if you have any questions, please post them in the chat and we will ask them all at once at the end. So, without further ado, may I introduce Dr. Craig Gelpi.
Thanks for that introduction. Some of those things sounded a little boring. So the Catalina Marine Society allows you to get in the ocean and get wet. That's a little bit more exciting. So the Catalina Marine Society is essentially a scientific charity chartered to increase our understanding of the ocean, local ocean outside LA. And we charity using volunteers mainly has a lot of advantages over other institutions in terms of cost, certainly, and also in duration of operation. So during this talk, I'm going to tell you some of the things we found really pertaining to Catalina Island. And at the end of the talk, I'll mention some of the other activities we have going on. Oh, my slide is not advancing. Okay, here we go. So as an introduction to the work, in 1992, 30 years ago, the Catalina Conservancy Divers, which is really a funding support arm of the Catalina Island Conservancy, like to dive, and they started measuring ocean temperatures at multiple sites and depths around the island. And they did so for many years. I could not find out why they chose to measure temperature, but it's a good thing to measure. Essentially, the heat capacity of water is very high. It takes a lot of energy in order to change the temperature of water. So if you have a sensor measuring temperature of the water and the temperature is changing fairly rapidly, you know that water of different temperature is passing over your sensor. So temperature is a good tracer for water movements in the short term. Not only that, but measuring temperature is cheap and relatively easy if you are a diver. In 2003, the nascent Catalina Marine Society began to analyze these data. And I'll show you some of the daily variation and annual variation, and we'll take a look at the phenomenology of that. But what we're going to show through many viewpoints is that Catalina really differs from the Northern Channel Islands in the mainland coast, and especially, of course, from Northern California. From our temperature studies, once we understood temperature, our studies expanded and we began looking at nutrients and phytoplankton. And phytoplankton, we use a proxy of chlorophyll. And we have some models that replicate what we see there. And now we're going on to study ocean pH in preparation for how ocean acidification may affect Catalina. But first, let me give you the oceanographic situation of the island. Okay, so here's a map of the LA area. You can see Long Beach and Catalina roughly just south of the center. Those big arrows on the left side of the plot are the California current, which California is oceanographically known for. It's a eastern boundary current, part of the North Pacific Gyre. We also have a lot of wind coming down the coast, and that wind generates a surface current. And as that surface current comes down the coast, the Earth is rotating underneath it, and water gets pushed off the coast to be replaced by cold, deep, nutrient-rich water off the side. That process is called Ekman transport. But that wind is interrupted by our transverse mountain ranges, like the Santa Monica Mountains. And so the wind doesn't make it into the Southern California bight in which the islands all lie. So we don't see that Ekman transport. We don't see that upwelling. In addition, past south of Point Conception, we have a recirculation on the California current, and that allows water that enters the bight to stay there relatively long time. It doesn't just fly by us on its way to the equator. It's around long enough to heat up and cool down and do other things. So that's the general situation. SoCal is just no NoCal. It's not Northern California in the least. Just scale the observed upwelling via satellite. I here have virtually the whole coast of California and an arrow indicating the California current. And we have satellite observations of surface temperature, blue being colder than red. And you can see off the coast of Northern California and Central California, the water is rather cold for this particular day in May. And it heats up as you get further offshore. That that is an example of this upwelling I was talking about. And if you see where Santa Catalina Island is, you can see the water is relatively warm there. The upwelling doesn't really occur in the bight. A lot of people don't understand this. It's perfectly obvious if you live down here. And it is a defining characteristic of the Southern California bight. Okay, on to some temperature measurements. I have two sets here. One on the left is 3 days of 30-minute temperature measurements at 4 different depths, 20, 40, 60, 80 feet. These were taken off of Wrigley at two harbors. Okay, they're color-coded such that the red line is the shallowest depth and the dark blue line is the deepest depth. And you see somewhat of a remarkable thing. At 20 feet, the red line is pretty much constant over these 3 days. There's some minor variation, but it's relatively constant. But if you look at the deeper depths, particularly you look at the blue line, the temperature at 80 feet, it changes quite a bit. It can change by 6 degrees in just a few hours, 6 degrees Celsius, so 10 degrees Fahrenheit. So you definitely notice this if you were diving. It changes about every 12 hours, corresponding roughly to the semi-diurnal astronomical tide, but not exactly. When I first saw data like this, I simply didn't believe it. And I had to query and pay attention to divers who were diving repetitively over many hours in the same location. And they could come back and tell me, yeah, the temperature did change by this many degrees. And when we thought that this was actually a real effect and not just an artifact of how the instruments were set out or set or whatever, then we got into analyzing the temperature data. So these variations are called internal waves, and they just represent large changes of water in the water column that simply damp out by the time they get to the surface. If you look at the right side, we have something totally different. We have, this is actually done by Karen Norris, who did a Herculean effort of taking data from all the sites around the island and combining them, averaging them so that we would get temperature as a function of depth for every hour for 10 years. And it's color coded the same way. Red is the shallow, in this case about 15 feet. The blue is the deep, in this case about 100 feet. And what you see plainly is a seasonal variation. Again, this is 10 years of data. So the seasonal variation is directly opposite the local variation, the local time variation. You get large swings of 6 degrees Celsius at the surface, while at 100 feet, the modulations are much smaller, just a couple of degrees. So intuitively, it's hard to put these two pictures together to get a consistent view. We know what's happening, but this tells you how the dynamics of the temperature are so interesting at Catalina. So, well, carrying further averaged over all the years, and this is the plot that you get. It just confirms what I just said. At the near surface, 15 feet, you're getting 6-degree change over the course of a year. The maximum temperature is around 21 degrees C, and it occurs somewhat in August. If you go to deeper depths, the temperature variation is only a couple of degrees, and its maximum is actually around 270, day 270. So a few months later than when we reach maximum at the surface. So I thought this was a revelation when I first saw it. Of course, dive boat captains were aware of this effect. They just never wrote it down and entered it into the scientific literature. These temperature variations may not mean much to you, so we'll have to compare it. We'll compare it to Monterey Bay temperatures. And so this is the surface temperature at Monterey Bay over the course of a year, given in a day of year like the other one was. And you can see first, the temperature is much colder in Monterey Bay. At the surface, the temperature is much lower than it is at 100 feet at Catalina. If you ever done any diving in Monterey Bay, you can appreciate the chilliness that you would encounter there. The other thing is that if I look on the left-hand side, the temperature starts increasing after the winter collapse of stratification at around day 90, at the end of March. If I look at the right side, around March, day 90, the temperature is actually decreasing, and it doesn't start increasing until just about the beginning of July. In fact, this is the upwelling season in Northern California. This is when the winds are coming down. If you stand at Point Reyes Lighthouse, with that wind blowing at you and you're watching those waves go by you, you can understand the upwelling season very well. That upwelling season really changes the whole nature of the curve that we see in terms of the seasonal variation at Monterey versus Southern California. So you can see, again, there are big differences in what we see at our island versus what we see elsewhere. Now, because the temperature maximum changes as a function of depth, it lags the deeper you go. As a physicist, you say, oh, that's due to diffusion. You have energy at the top of the ocean is diffusing down to lower depths. So maybe I can calculate what the diffusion is. Maybe I can make a model of why the temperature behaves this way. And that's what we did. We we understand the amount of sunlight, shortwave radiation coming through the atmosphere and entering the ocean. We understand the evaporative cooling. We understand radiational cooling in the long wave. We understand changes of energy between the atmosphere and the ocean just due to sensible heat and a few other things. And we understand the propagation of photons through the water column and how they warm the water column as a function of depth. And when you put all that together, there's only one thing you need in order to give you the temperature as a function of depth, and that is called the vertical diffusion coefficient. And so we put this model together, we fit for the vertical diffusion coefficient, and we take the model and then we can draw these black lines that you see in the plot, and they match the data rather well. The only free parameter here is the vertical diffusion coefficient. And it's a very simple number to remember. You can round it off to 1 centimeter per second. And it's a fundamental number to have in the ocean. And this is one of the reasons why it's so good. Just reminding you of ocean marine biology 101, we know that the sunlight is coming through the surface and phytoplankton gather there to get the sunlight. And so they use up the nutrients at the surface, they grow, they multiply, they form the basis of the food web. Okay, they're eaten, they're consumed by various marine fauna, which eventually die and decay and fall down to the seabed. They're remineralized by bacteria, which releases carbon dioxide and nitrates and nitrites, and they therefore leave a reservoir of nutrients on the seabed. So the phytoplankton have depleted the nutrients at the surface. There's a lot of nutrients at the seabed. It's a natural mechanism for nutrients to diffuse up to the surface. Now, diffusion in the ocean is different than diffusion as we normally think about it. It's not molecular diffusion, excuse me, where molecules are changing places, or heat diffusion, where a fast molecule is colliding with a slower one and giving it some of its kinetic energy. No, diffusion in the ocean is driven by mixing of parcels of water, parcels of various scale size. So any characteristic of a parcel of water as it mixes, all the things that are in that parcel of water will mix at the same rate. So this is why this is diffusing the heat coming down. It's this eddy diffusion is what we call it. And it's the same mechanism by which the nutrients diffuse up to the surface. So by studying the transfer of heat, we understand the rate, if we have the profile of nutrients, the rate at which nutrients are delivered to the phytoplankton. And marine biologists can calculate this another way. They can look at the profile of nutrients and they can see how fast the phytoplankton grow, and they can estimate, well, what must be the diffusion in order to feed those phytoplankton. It turns out that the two numbers are the same. We get the same number using the physical properties of temperature that they do looking at the growth of phytoplankton at the surface. But yet there's more. Our climate is determined by a thermohaline circulation, which essentially redistributes energy around the ocean. This is a standard cartoon. Blue represents cold water flows, red represents uh, high temperature flows. What happens is that the ocean is chilled and becomes dense at the surface in the Arctic Ocean between Greenland and Iceland in that area. The chilled, dense water sinks to the bottom and eventually floods all the ocean basins. And this is the reason why water is cold anywhere you go if you go deep enough in the ocean. Conservation of water dictates that that cold water has to come back up to the surface. And it does through in some diffusive manner. And many ocean physicists have looked at the process by what goes in, what comes out. They say, well, the diffusion value must be 1 centimeter squared per second. Is that a coincidence? Perhaps. But we have proposed the Catalina be a natural laboratory to study diffusion because one of the diffusing mechanisms in a deep ocean is thought to be internal waves. And of course, Catalina is an internal wave hotbed. So I would certainly like to proceed on this problem. I don't know if I ever have time to, but we hope that others will, and see if we can understand more about global climate looking at local processes at Catalina. Okay, so a one-dimensional temperature model, by one-dimensional, I mean just vertical only, but it's also in time, has been very successful. And we could imply that one-dimensional temperature model because the diffusive processes work for various characteristics. We want to try to understand the nutrients going to phytoplankton, and we can look at phytoplankton as proxy by chlorophyll, which we can measure through its pigment. So perhaps a simple chlorophyll model will also work in the Southern California Bight. We do know that due to little upwelling in the diffusion coefficient that we calculated, we can expect very little chlorophyll in the center of the bight. So we went and we looked at satellite imagery, satellite spectral imagery for 10 years. We got the data globally, but we emphasized the Southern California Bight. Here's an average of those data. Essentially, the satellites collected in a bunch of color bands, wavelength bands, and it's possible to estimate a chlorophyll product by just simply, essentially how green the water is after you do some other things that compensate for, excuse me, atmospheric propagation. So now I have this map of chlorophyll. The darkest blue just represents land masses in which there is no chlorophyll. I have two boxes drawn, one around Catalina and the other around San Nick. And you can see there's relatively little chlorophyll, just light blue around Catalina. If you look at the Northern Channel Islands, there's lots of chlorophyll. It really emphasizes red here. but it's about 10 times as much as we have at Catalina. That's rather surprising because you think if you go to the Northern Channel Islands, you would be able to see a difference in some way, but I've never noticed the difference, either diving in terms of the amount of life or the color of the water. But these 10 years of satellite data, hundreds of data sets. The other interesting thing is that if you look at San Nick, there's a lot of chlorophyll around San Nick. And we looked at this quite a long time. We tried to figure out if water is just, if high chlorophyll content water was coming from the Northern Channel Islands. We could never prove that. And then it finally occurred to us, and actually, people at Scripps were working this same problem at the same time. They came up with the same suggestion, is that there's a ridge called the Santa Rosa Ridge. It extends from Santa Rosa Island to San Nicolas Island, which the water is quite shallow. If you're familiar with Beg Rock is on this ridge. So this ridge is like 70 meters, is a good scale length. So if there's any current on the seabed, it's pushing the water. It has to push up highly nutrient water that's hundreds of meters deep up over the 70-meter depth ridge. Of course, then the nutrients get to the sunlight and it produces phytoplankton, which lead to the chlorophyll. So even though we have another southern Channel Island, it is quite different in Catalina. We expect that San Clemente is very similar to Catalina in terms of all these aspects, but we don't have any data there. Now we move on to ocean acidification. Okay, well, I'll explain ocean acidification if you're not familiar with it. Essentially, carbon dioxide, as we know from high school chemistry, uh, interacts with water, or at least diffuses into water very rapidly. One reason why it diffuses rapidly is because once it gets in the water, it undergoes some chemical reactions, producing less carbon dioxide, so it's willing to take in more from the atmosphere. So when CO2 interacts with water, it produces carbonic acid. So wherever you have CO2, you're going to have carbonic acid. There are two major sources of CO2. One is at the surface, and this is due to atmospheric CO2, and another one is at the seabed, which is produced by the decay of dead organisms, as I discussed earlier. You also get some respiration at the phytoplankton layers, which are fairly shallow, but those turn out to be relatively small given these two sources. The source at the seabed is by far the greater source. So you would have a lot of carbonic acid closer to the seabed. In fact, you can make this little cartoon plot I have to the left. Its vertical axis is depth, going from the surface to deep, and the pH will indicate increasing acidity as you go to deeper depths. But for ocean acidification, what happens is that CO2 increasing in the atmosphere means that we're going to put more CO2 in the ocean, which is going to turn to more carbonic acid, and essentially we're going to have an ocean that's basically is going to go more toward a neutral state, and we call this ocean acidification. And this changes the water chemistry quite a bit, and it may have a severe effect on a lot of marine fauna. So we want to understand this. Now, our organization cannot fund the state-of-the-art instruments to measure the exact increase in pH due to increasing CO2. But what we can do is we can understand the dynamics of the water and how that would influence measurements that are made. And that's what we have here. Our program is called the Catalina Dynamic Ocean Chemistry, called CDOC, as in sea physician, so to speak. And here we have a picture where we have divers deploying a sonde, which is essentially an underwater computer with sensors attached to a mooring chain outside of Wrigley Marine Science Center. So if you go out there, you'll see a platform that's moored out in the cove. And that cove, that mooring really supports instruments that we put out there to do time series of ocean chemistry. We have other components to CDOC. namely volunteer boaters can take their boat out there. We'll meet them at the dock. We'll give them a sign for them to deploy into the water column so we can get water column measurements. The sign that was deployed in the previous chart was at 60 feet. So it's only getting a time series at 60 feet. But here we have a kayaker who's going to put the instrument down to 100 feet, stopping every 20 feet on the way down to take data, and every 20 feet on the way up. And we also have divers who can take a sond down. We have a special sling that they can wear such that the instrument won't interfere with their gauges. And they can dive in the park down to 80 feet, as far as you can really go in the park and stay in the park, stopping every 20 feet and collecting data. We collect data about every 5 seconds. So we have a lot of water profiles, and we have a lot of averages from 60 feet of pH and other water chemistry um, constituents. And a lot of work is going into this, but I'll just show the comparison of our pH to what other researchers have found throughout the bight. And by throughout the bight, this is La Jolla Kelp Forest, San Miguel, Santa Cruz, Anacapa, and also a mooring off of Malibu. And what we find is that for those measurements made at the Northern Channel Islands and right on the coast, an average value of pH and pH units is about 7.9. These have all been extrapolated so that they're all at the same depth and extrapolate to any other thing that we could do to equilibrate the measurement. If you look at what Catalina is finding, what we find at Catalina and what we find in deep water Malibu, deep water being several thousand meters, I think, we get a value that's around 8.1, which is significantly different. So Catalina, put in the vernacular, is less acidic than all these other places, which itself was never a complete surprise to us. We didn't know what to expect, but we didn't expect it to be really different like this. So we're getting time series. I mean, I just showed you the averages, so things sort of the overall big picture. But we can look at the time series. Again, when I showed the temperature time series, I showed a period of it earlier. But this is how things are dynamic, excuse me, at both temperature and pH at Catalina. Again, you see the internal waves at the top. We have temperature at 4 different depths. We have pH at 60 feet in the bottom panel. And you can see that it's modulated quite a bit. It's modulated as the temperature is modulated. And these modulations are actually much larger than the decrease in pH we've seen over the last 100 years. So you have to understand these modulations in order to interpret your measurements correctly, and especially those pertaining to how fast is the ocean acidifying. Now, the other thing you might want to notice here is the pH measurements seem to be flat topped. As a sensor guy, I look at this and say, oh, my electronics are saturated. Yeah, I have to make some adjustment or take that into account. Well, that's not really what's happening. The electronics aren't saturating, the ocean is saturating. So when the internal waves, as you can see at the top, the temperature at 80 feet at sometimes is equal to the temperature at 20 feet. And the only way that this can happen is if the water at 20 feet came down to 80 feet. Therefore, they would have the whole temperature, and the whole water column would have the same temperature. It's pretty hard to imagine, but yet that happens because of the island slope. Excuse me. When the mixed layer comes down to 60 feet level, there's no way to increase the pH anymore because you're in the mixed layer. So that gives you this apparent saturation effect. Seems like a minor point, but when you think it through, it can have some importance. So y'all been a pretty good audience. Let me give you a speculation so that you have food for the mind. First, let me say that the island internal waves, this is what it looks like. If you look at the ocean, we have the mixed layer with a temperature gradient of 0, and below the mixed layer, below the thermocline, if you're diving, you have a temperature gradient and a pH gradient. The two are highly correlated. And if you had some modulations in the stratification, say the black box being an instrument um, site, you would just measure those. However, when you have big internal waves, you look at the right side of this, and the water flow is distorted due to the slope of the island. And you can actually get into the mixed layer through this distortion. And when you do, you see that flat top to the pH profiles that we just looked at. So the speculation now is what is the future of average measurements given climate change and given ocean acidification? So this is a cartoon. The vertical axis is depth again. The horizontal axis can be temperature or pH. If you look at the black solid line, that represent the sort of the average current temperature profile. You have a vertical line at top, which is essentially the mixed layer, and then the temperature fares into the deep ocean temperature as you have a negative positive gradient going downward. Um, that's just typically what we see. Now, with global warming, climate change, we have more energy in the ocean. We're going to have higher winds. We're going to have higher waves. We're going to have enhanced mixing. So our mixed layer is going to get deeper, and that's shown by the dotted line. Our future temperature profile is going to have a deeper mixed layer, and it's going to increase. I mean, temperature throughout the ocean is going to increase, but you can see the trend there to deeper depths too. Now, the interesting thing is you look on the left-hand side, the red curve, the current pH has a mixed layer, same mixed layer that the temperature has now, and of course, then a gradient going down to the seabed. But with ocean acidification, CO2 is increasing, we're increasing acidity at the surface. So the value of pH at the mixed layer is going to go to the right, to the left. All right, you're decreasing pH, you become more acidic. However, because you're increasing the mixed layer depth, there may be regions, like right here, where even though you're decreasing pH generally everywhere, there's going to be regions where the pH is not going to change much, or indeed, if it's very severe mixed layer increase, the pH could actually increase. Now, if you're looking at the effect on marine fauna, they're going to try to find uh, ocean acidification refugia. And places like Catalina may be that. There may be depths in which the pH changes very little because you have these two competing effects. So I have a few more slides. Let me just summarize what we've seen. That Catalina is really a different place. You have higher temperatures, higher pH, less phytoplankton there relative to the northern islands and even San Nick. This is all indicative of less upwelling due to sheltering of the local winds and greater downwelling due to the internal waves on the island slope. And looking back at this, it should have been obvious. There's no reason to do this work. We should have realized this 20 years ago. But of course we didn't. Back then it was all a big mystery. These aren't the only projects we have. I'll just run down some that we have just so you can understand the organization a bit more. We have the David Sile Continental Thermograph Array, and this is a series of thermographs that go from the LA Ventura County line down to Laguna Beach. And we've been collecting data there for about 15 years. We recently deployed last year, right after the pandemic allowed us to, the Jim Updike Seabed Station. And you can see a picture of the seabed station at the top right. It actually measures current at the seabed. There's some things we can't measure on a mooring chain. You need a stationary platform. And this concrete block situated at 100 feet depth holds a variety of instruments as well as a current meter. Unfortunately, but we have got some good data. We are ending our Monterey Bay thermograph array. And the end result is we're going to take data, particularly internal wave temperature data at Monterey, and we're going to compare it to what we see in SoCal. We are heavily involved in what's now called the Remotely Determined Ocean Conditions Project, in which we're trying to estimate wave height as well, wave direction is pretty easy from sequences of JPEGs obtained from the camera. The idea here is that you may get very local conditions in local coves, or this was particularly determined by Monastery Beach in Monterey. You want to understand the wave conditions that could produce dangerous conditions or that have other influences that you may want to seek for a scientific purpose. we've been getting our T being temperature, where we're trying to figure out how deep do these internal waves influence things. So we think they could be several hundred feet deep. Scuba divers can't get down there very easily, so we developing techniques and have acquired the sensors to begin our deep T project. And the latest thing we're doing is we're planning to make a mixed layer depth seasonal model. So that speculation I talked about, we can just put more meat on it to see what it really means in terms of climate change, mixed layer depth, increasing temperatures, decreasing pH, etc. And finally, some acknowledgments and references. Of course, we have help from the Aquarium of the Pacific, California Science Center. We get a lot of data from the Channel Islands National Park. We get data from clubs like the American Legion Yacht Club in Newport. The Antelope Valley Desert Divers have been supplying us with temperature data from Ventura state line for a while. Initial data came from the Catalina Island Conservancy and, of course, the universities and government organizations, which make their data freely available to you. There's a list of papers here. I'll give them the technical details of the things that I described, except for the speculation. But the top paper is describing our organization. And we think our organization can be a blueprint for worldwide organization, well, not world, other organizations throughout the globe, because we need people to gather data so we know how to compensate for climate change and ocean acidification as it's upon us. So that's all I have, and certainly can entertain questions.
Outstanding. Thank you very much, Craig. Certainly do appreciate it. I was actually going to turn it over to Tiffany, who has been monitoring the chat here. And I do see that, I asked a few questions in there. So, but I just wanted to remind the audience, if you have any just burning questions about oceanography around Catalina, please feel free to put it into the chat. Oh, it looks like there's a few more. Okay. Tiffany, you want to take over here?
Yeah. All right. Thank you, Craig, so much. Well, I'll go. You're getting kudos in the chat, by the way, from people who are saying it's a very interesting talk. But question number one. So the tongue of upwell cold water that you showed that engulfs San Nicolas Island, how does that change seasonally? And will less nutrient concentration around Catalina comparatively? How can we account for the thick kelp beds that we see around Catalina?
Well, the higher concentration of phytoplankton around San Nicolas is there almost all year. It dies back down in the summer somewhat, but we looked at it month by month. It's actually all those images on one of the papers in the references. So it's an outstanding feature.
Cool.
Nutrients are less because we see less phytoplankton at Catalina, but apparently there's plenty there for the kelp. Like I say, diving, I really haven't noticed a difference in the marine fauna or flora from the Northern Channel Islands to Catalina. So I can't explain why it's so rich. I'm glad it is. But as a little experiment for yourself, the next time you take a ferry out to the island, you come back, before you leave, fix your eyes on the water, try to memorize how blue that water is. And as you come closer to San Pedro or Long Beach, not right adjacent to it, but several miles out, you will notice how greener the water is. There's just a lot more phytoplankton close to the coast. Maybe that's due to pollution or whatever, I don't know, but it's there. But I can't explain why the island is so rich in life. Just glad it is.
You're making me want to come out to the ocean. I'm landlocked in Nevada. All right. Question number two. Given your speculations on the future of pH in our offshore environment, How do you suspect the marine life around Catalina might respond to the presence of developing pH refugia relative to surrounding areas where pH may actually end up decreasing?
Yeah, we think Catalina may become well, we said it was a refugia. Uh, that may be the last remaining place that pH sensitive uh, life forms may be able to hang out in the bite. I mean, we talked, everything may be able to adapt as pH changes. But if they can adapt, they're going to be at San Clemente and Catalina Island. That'll be the last places that they'll be able to live. And this
Wow.
includes the oysters, but we don't have oysters in Catalina, but any mussel, shellfish, things of that nature. Uh, we ought to be concerned and we ought to think about where we would locate marine reserves in order to protect against future acidification threats.
Sure. All right, I'm going to save that third question as a wrap-up question. So from Douglas Pace, he says, great talk. Given the need to increase monitoring throughout the world, are there other locations that have oceanographic conditions similar to Catalina that would be valuable for comparison?
What are some that don't? I don't know. The thing that makes Catalina sort of unique is it's in a sheltered environment, but it's a real ocean environment. So we have tried to put together partnerships with Palau, people in Palau who collect temperatures, but that hasn't worked out. But we would certainly like to see what's happening there. I mean, they have a lot of rainstorms and squalls, but on the other hand, it's a fairly quiet place compared to other islands in the Pacific. Um, Other than that, no, I wish I knew more about the global climatology of islands, but I don't. Oh, Easter Island would be one. Easter Island, it gets a little not too rough there. They don't have a lot of hurricanes or whatever, and they're known not to be an upwelling center. So you don't have a lot of life in Easter Island. And you can refer to the Smilodon if you want to read a little bit more about that.
Yes, Craig also takes care of the Smilodon if you received those in your email. So thank you, Craig, for that. So we have another question from Vivini Oviedo. Thank you, Craig, for the talk. In Santa Catalina Island, are specific locations that tend to be warm or cool water? I'll repeat that. In Santa Catalina Island, are there specific locations that tend to be warm or cool water? Through the years of recording the temperature, did you notice a change of the number of species in Catalina Island, maybe as a result of those differences?
No, we haven't been able to correlate that to the species counts. The Catalina Island Conservancy have been counting, they've been counting kelp and other species in abundances for a while. We do note that on the southern end of the island tends to be a bit warmer because it tends to be in a wind shadow. And in a wind shadow, you have less mixing. And that means the surface water is going to be warmer. But that's well documented in the literature, not by us.
Got it. All right. Are there any more questions for Craig before I we got a technical question, he says. What is the depth limit for satellite measurements of chlorophyll? Are there technical or computational advantages that can allow for greater understanding of primary productive changes at greater depths?
Well, that's a problem with remote sensing. Visible light, which is what you need to see green pigment, you can't tell where in the water column. I mean, you have to integrate all light in the water column to see, what's the total effect. And you really can't do that very well. I mean, your sensor integrates it for you, but you don't know what depth it's come from. So these chlorophyll tools that we use in remote sensing have been derived empirically by people measuring chlorophyll and then looking at how it maps to the greenness of the water as the satellite determines it. That's the first part of the question. I missed the second part of the question.
Yeah, I can read it again. Are there technical or computational advances that can allow for greater understanding of primary productive changes at greater depths?
Yeah, the way you can get out there from radiated transport, which is my expertise, is to use active light sources. So this is done so much. You shine a laser into the water and you see it backscattered, but you can time how long it takes for that return to come back. And that allows you to get an estimate of the backscatter as a function of depth. I think it would be very, very useful. And I'm sure it must be done in places now. So that way you can look at the amount of chlorophyll, but you could also look for other nutrients like that that have some uh, optical response. And then you can measure things happening as a function of depth, which I think would be a very interesting physics problem and probably a very useful biological tool.
All right. He says, as a larval biologist, I want to be able to know what food is out there for my larvae. So, yep. All
Well,
right.
people are working that issue with hyperspectral measurements of the ocean. So they try to determine the various species of phytoplankton that are out there by fine changes in the spectral response. So that's actually a very hot area of research. I just came back from the Ocean Optics Conference, and there was a lot of that going on, trying to understand optically what the species are that you're looking at.
Hmm. Interesting. I guess I have a question. I deal more with things on land. How much of this that's used for the ocean can be transposed to our plants on land with different spectral I don't know the
Oh,
right
quite a bit.
term. Yeah,
Uh, Observations of plants on land are far exceed observations of the ocean.
I see.
And the reason is that the land is very bright. And so you
Mm,
don't have to, the atmosphere doesn't influence you. If you look from a satellite at the ocean, 90% of the photons that you get come from the atmosphere and 10% from
mm,
the ocean. If you look over land, it's just the opposite. So you got a much stronger signal. And a lot of people are working to try to understand uh, detailed characteristics of plant cells on the surface.
thank you. All right. Are there any more questions before I kind of end with a wrap up question? Anybody else? Going once, going twice. All right, so our last question for you, Craig. Thank you so much for this talk is how can students and other volunteers with the Catalina or get involved with the Catalina Marine Society? What sort of things can they do? What do your volunteers do? How can they get involved?
Well, you can go to our website and see what we do. It is also a contact link, so you can contact us directly, which would usually be me. Uh, we have a big need for data analysts. And by data analysts, I mean all all levels of analysis. It could be first just making plots. I mean, like I said, we
Mm,
had 15 years of temperature data from Laguna Beach. We'd like to see how that data is related to the data at Shaw's Cove, just a little further down. Simple things of learning signal processing, doing correlations between time series, making the plots. Those are all that new people who are interested in science, interested in the ocean could do. And then again, we have researchers at JPL with PhDs that contribute some time to us, but they have a lot of domain knowledge and a lot of training with special tools that they can use. So, and then there's a lot of field work. We have a lot of people put out thermographs. They're very easy to do. We only ask that you know the area very well so that if you put out an instrument, you can go back and get it. We tend to, we don't want to lose them. We lose quite a bit. That's the cost of doing business, but we, we like to think we get them back.
okay. Well, I'll see if I can throw that link to the society in the group chat before we log off. But thank you so much for being here. Mia, do you want to thank you as
Sure.
well?
Thank Craig. Great talk. Super interesting. Even being very familiar with Catalina, this is like a different angle of how you presented. That's not my expertise, so it's really cool. So thank you very much. For those of you still here in the audience, just please check back for updates on our website about upcoming talks as well as opportunities that Chemo highlighted at the beginning for both funding and also different research opportunities for high school students and other things for possibly college and graduate students as well. And hope to see many of you at our annual conference at UC Irvine in April. So thank you very much and hope to see you at our next Scas Spotlight. Have a good night.
Good night.